Hey everyone, and welcome to this explainer. Today, we're diving into what is arguably one of the most transformative techniques in all of modern biological research. If you've ever been totally mesmerized by the sheer, staggering potential of genome-wide genetic analysis, well, you are exactly in the right place. We're going to break down how scientists today are rapidly unraveling the complexities of the human genome and pinpointing the drivers of disease with just unprecedented precision. But first, let me throw a question your way. How on earth do you find one crucial gene, hidden among literally billions of base pairs? For the longest time in genomics, trying to figure out which specific genetic sequence was responsible for a specific cellular behavior, it felt exactly like trying to find a needle in a haystack. But what if, instead of meticulously sifting through all that hay by hand, you could just bring in a giant magnet? Well, that is exactly what CRISPR screening does for genomics, and it is completely revolutionizing how we approach genetic discovery. Okay, let's dive into this. Here is our roadmap for today. We're going to cover the power of CRISPR, how CRISPR screening works, unlocking modern biological mysteries, partnering with CD Genomics, real-world case studies, and finally, how to accelerate your next breakthrough.
All right, part 1, the power of CRISPR.
To really appreciate where we're going with this, we've got to quickly look at where the technology actually comes from. Out in nature, the CRISPR/Cas system evolved as this highly effective immune defense mechanism in bacteria and archaea. It was basically designed to recognize and chop up the invading nucleic acids of bacteriophages. And today, we've harnessed that exact same precision for the lab. Put simply, CRISPR screening is a large-scale genetic loss-of-function approach. So instead of just editing one single gene to see what happens, we generate and screen an entire population of mutant cells at once. The brilliant idea here is to systematically knock out every single gene in the genome that could potentially be important. But, and this is absolutely crucial, only one gene per cell. It's like a massive, parallel investigation into genetic function.
Now, you might be thinking, wait, haven't we been doing large-scale screening for a while? Well, yeah, we have, but CRISPR completely outclasses the older techniques. Just take traditional RNA interference, or RNAi screening. It's historically struggled with low transfection efficiency, and it could only temporarily suppress gene expression at the mRNA level. Kind of a half measure, right? On the flip side, high-throughput CRISPR/Cas9 screening gives you permanent gene editing. It's robust, it's highly programmable, and it offers a level of efficacy that makes it an absolutely indispensable tool for precision medicine today.
Moving on to part 2, how CRISPR screening works.
So we know what it is, but how do you actually pull off a genome-wide screen in the real world? Well, the crucial point is how these 4 stages seamlessly connect. Step 1 is constructing the computationally designed library of single-guide RNAs, or sgRNAs, which get synthesized and cloned into a delivery vector. Step 2 is the screening itself. This is where you introduce those components into your cells and apply selective pressure. Next, we shift from physical lab work to the data side. Step 3 leverages next-generation sequencing, or NGS, to read the results. And finally, step 4 is measurement and bioinformatic analysis, where those sgRNAs are recovered and linked right back to their target genes. Now, during that second step, the physical sorting of cells post-transfection, you're basically relying on one of 2 primary methodologies. You can run a positive selection screen, which is all about identifying genes whose disruption actually grants the cell a growth advantage or resistance under really harsh conditions. Think of it as finding the survivalist. Or you can run a negative selection screen, which is designed to find genes whose disruption causes the cell to die off or become super sensitive to treatments. Both routes give you incredibly powerful yet distinct biological insights.
Which brings us to part 3, unlocking modern biological mysteries.
Let's look at how this builds into actual lab applications, because the sheer scale of what this technology can achieve is just breathtaking. By isolating exactly which gene knockouts yield relevant phenotypes, researchers are discovering vital disease-related genes and entirely new targets for drugs. They're establishing phenomenally accurate disease models. But what's really mind-blowing is that this technique is now being applied to study the 98% of the human genome made up of non-coding sequences. You know the stuff we used to call junk DNA? Turns out that junk plays absolutely pivotal roles in gene regulation and tumorigenesis. People are even combining this with single-cell sequencing to map out wildly complex biological regulatory networks.
And that leads us right into part 4, partnering with CD Genomics.
Look, as powerful as this science is, executing a flawless, high-throughput screen with deep sequencing is incredibly complex, data-heavy work. For most labs, success really hinges on finding a reliable expert partner to shoulder the computational and sequencing heavy lifting. That's exactly where partnering with CD Genomics comes in. They've developed this highly optimized, robust strategy that offers affordable, reliable, and high-throughput CRISPR screen sequencing using amplicon-based NGS. Basically, they act as the ultimate guide for your lab. They provide direct, hyper-detailed data on sgRNA abundance, targeted gene analysis, and functional enrichment. They take what could be a generic, clunky workflow and transform it into a finely tuned pipeline.
The specific advantages they bring to the table are completely designed to save you time and budget. With their pipeline, you get to bypass those super laborious, time-sucking cloning steps entirely. You also get dedicated, one-on-one support from specialized PhD-level scientists who oversee your project. They're utilizing industry-leading Illumina HiSeq platforms for paired-end sequencing, running the bioinformatics through the advanced MAGIC tool, and they offer a ton of multiplexing flexibility. The end result? Cost-effective, unbiased, and highly sensitive detection.
And getting this integrated into your current workflow is shockingly straightforward. If you're submitting a DNA sample post-selection, you literally only need about 1.5 micrograms, with a concentration of at least 30 nanograms per microliter. Or, if you're working with cell samples, a baseline of just 5 million cells is all it takes to kick off the entire deep sequencing and analysis pipeline. It's concrete, it's accessible, and it is totally ready for your lab's next big push.
Part 5. Real-world case studies.
Now, if you want to see how these precise services actually translate into publishable, real-world results, let's look at this brilliant study from the Journal of Experimental and Clinical Cancer Research. The team here was investigating cancer metastasis, specifically how tumor cells develop resistance to a type of cell death called anoikis. To figure this out, they ran a pooled genome-wide CRISPR-Cas9 screen using the Gecko V2 library in SKOV3 ovarian cancer cells, aiming to hunt down the exact genetic drivers behind this deadly resistance.
Following this screening under both standard and ultra-low attachment conditions, the high-throughput sequencing data came back, and it was staggering. It pinpointed exactly 286 genes from negative selection and 122 genes from positive selection that were directly involved. And while the screen successfully found some known resistance genes, it really shined a massive spotlight on one specific, critical target, the PCMT1 gene.
And this brilliantly illustrates how a CRISPR screening pipeline uncovers direct causal relationships. The data paints this perfect narrative arc. Step 1. The CRISPR screen flags PCMT1 as essential for anoikis resistance. Step 2. Further IPMS analysis confirms that PCMT1 directly interacts with the extracellular matrix protein LAM3. Step 3. Functional studies prove that silencing LAM3 visibly reduces cell spheroid formation and migration. Boom. Cause and effect.
So, what does this all mean? The researchers concluded that PCMT1 upregulates FAK-SRC signaling to actively promote metastasis. Because of the precise genomic targeting made possible by that initial CRISPR screen, we now know that targeting PCMT1 with specific antibodies actually shows genuine promise as a therapeutic strategy for fighting ovarian cancer metastasis. Guys, that is a real, tangible medical breakthrough, and it was born directly from high-throughput screening.
Finally, Part 6. Accelerate your next breakthrough. We've seen the mechanics, the massive scale, and the stunning real-world applications of this technology. Now the focus turns directly to you and your own lab. The genome holds the answers to some of the most complex diseases humanity faces, but uncovering them requires scale, it requires precision, and it requires having the right strategic partners in your corner. So I'll leave you with this one final thought. What will your next genetic breakthrough be? By letting experts like CD Genomics handle the heavy lifting of next-generation sequencing and bioinformatics, you're freed up to do what you actually do best: design the experiments, interpret the biology, and discover the future of medicine. Thanks so much for joining me for this explainer, and hey, happy researching.